Okay, hello everybody again. So today's video is going to be about DNA. So we are going to title our notes DNA. And we've talked about DNA before. And so if you remember that this word, DNA, stands for a long scientific word. Do you remember? It stands for deoxyribose nucleic acid. And so it is a nucleic acid that is one of the major organic molecules that we find in all living things, and DNA is super important. And if you remember from the past videos, we are all made of cells. All living things are made of cells, and all cells, well, all eukaryotic cells have an organelle called a nucleus, and inside the nucleus is where the DNA lives. Okay, and so today we're going to focus on DNA, what it looks like, and what it's made of. And I have talked about this in the past, but I'm going to go into a little bit more detail today. Okay, so DNA is made of monomers like all big biomolecules are. And the monomer for DNA is called a nucleotide. And if you remember, nucleotides are made of three parts. Okay, so nucleotides are made of three parts. And the three parts are the following. We're going to write a list and we're going to draw it. And we've done this before. The first part is a phosphate group. And I'm just going to draw that as a P with a circle around it. The second part is called a sugar. And for DNA, the sugar is a deoxyribose. Oops. And so let me scoot over so we could draw that. And so, oh, sorry. This board's a little sensitive today. Sorry about that. Okay, so here's our nucleotide, and the phosphate group is attached to a carbon. So there's a carbon here. That carbon is attached to another carbon. This is attached to an oxygen. This carbon is attached to another carbon. This carbon is attached to another carbon, and that's attached to the oxygen. So this is the sugar, which is a deoxyribose sugar. And then the third part is a nitrogenous base. And the nitrogenous base is attached to this carbon. And for now, I'm just going to put an A. And there are four choices for the nitrogenous bases. There is A for adenine. There is C for cytosine. T for thymine. And the other one is G for guanine. Okay, so those are the four choices. And so a couple of things I want to point out that you need to know now. So we've talked about this before. Um, the first thing that I want to point out is that these carbons in the sugar are numbered. This carbon here is carbon number 1. I'm actually going to put the number 1 here. This carbon is carbon number 2. This carbon is carbon number 3. This carbon is number 4, and this carbon is number 5. Okay, and that is important because each nucleotide has a top and a bottom. And the top is always designated as the 5' end, and the bottom is designated as the 3' end. And that is because the fifth carbon is up on the top and the third carbon is on the bottom. Okay, so let me just circle that. Here is the fifth carbon, so that's the top, and here is the third carbon, there's the bottom. And so each nucleotide is attached to another nucleotide, attached to another nucleotide, and a DNA molecule are thousands of nucleotides attached to each other. And there's a top and a bottom for each nucleotide and for the whole strand of DNA. Okay, I'll explain in a second. Okay, but before I go into that, I just want to point out there are a couple of things that you need to know. One is that there are these four choices, A, C, T, and G. These are all called bases or a nitrogenous base. And what you need to know is that adenine, A, always binds with T, and cytosine always binds with G, okay? And vice versa. If you have a T, it binds with A. If you have a G, it binds with C. So A and T always go together, and C and G always go together. And so what I want to point out is that these bases, they are actually molecules. Okay, so I'm going to pull in a picture of these bases so you can see what they look like. Hang on a second. Okay, so here is a nucleotide. And so I'm going to show you a cartoon picture of what I just drew. So I'm going to scoot it over here. Actually, I'm going to move it over here so you can see. Okay, so here is my drawing, and here is a cartoon picture from your book. And so here is the phosphate group, and so there's the phosphate with some oxygens and hydrogens attached to it. Here's the deoxyribose sugar, and so you can see there's an oxygen here, but each of these corners represents a carbon. They're each numbered. And then this is the base. And so this could be A, C, T, or G. But I want to point out that A and G are called purines, and C and T are called pyrimidines. Okay, I'm going to write that down, but let me first show you a picture so you can see what I'm talking about. Okay, so here is a picture. Let me scoot that down, and I'm going to scoot up. So these are the bases that are possible. Let me enlarge it just a little bit. It's a little blurry, sorry. Okay, so what you need to know is that the pyrimidines, so write this down in your notes. This is how it's spelled. Pyrimidines are C and T. Okay, so here's C and T. There is this other one called U, which is RNA, which we'll talk about in another video. And then there are purines. And the purines are A and G. Okay, so the way that I remember these is that I remember A and G are pure, and I remember it as angels of God are pure. And then I remember the other ones are the pyrimidines. Okay, so when you're asked, which ones are purines, they are always A and G, and A and G always have two rings. See here how A has two rings? There's a 5-carbon ring attached, fused to a 6-carbon ring. Guanine is a 5-carbon ring attached to a 6-carbon ring. So these purines have two rings. So I'm just going to write that here. There's two rings. And then a pyrimidine only has one ring. So see here, cytosine has one ring. It's a 6-carbon ring. Thymine is a 6-carbon ring. And uracil is also a 6-carbon ring. You don't have to know what uracil is right this moment, but it is a pyrimidine. So here's the rings. This C and T are one ring. So C and T are a one-ring molecule. Purines are a two-ring molecule. And this is why. Okay, I'm going to scoot up. So I'm going to redraw a nucleotide, okay? Draw with me. So here is a phosphate group. I'm going to go a little bit fast, and I'm going to draw them a little bit easier than before because I'm going to draw several of these. So here is one nucleotide. I'm going to say this is an adenine. And remember, an adenine is a purine, and purines have two rings, okay? Let me just check, make sure that's right. Purine has two rings, yeah. Okay, then the next nucleotide gets bound through a dehydration reaction. Okay, so we're building bigger molecules, And the phosphate group to the next nucleotide is bound to this third carbon here. And so here is the next nucleotide. So here is the sugar, and then I'm just going to put a C here, okay? Then I'm going to draw another nucleotide. Another nucleotide is bound here. Okay, so I'm just going to draw 3 of these. And then I'm just going to put a G, okay? So remember, the A's and the G's have 2 rings. The C has 1 ring because it's a pyrimidine. Okay, so far we're building a DNA strand. This top part is the 5' end, and this bottom part is the 3' end. Okay, there's an orientation, there's a top and a bottom, just like our bodies. We have a top, which is our head, bottom, which is our feet. Then for DNA, it normally looks like, when you see DNA, it looks like a double helix, right? Let me just draw it like this. Okay, and so what we're doing is we're drawing this double helix. And so if you know one side of the double helix, then you automatically know the other side. And like I said, A always binds with T. So I'm going to draw an upside down T here. Actually, let me change colors. So I'm going to draw another, let's do orange. A is going to bind to T. And so I want to point out that A is a doubled ring molecule and T is a single ringed molecule. Okay, let me just color code this. So there's 2 rings here, and then T is a 1 ring, okay? Oops. Okay. And so the T is bound to a sugar, and I'm going to draw the sugar. It's upside down. And here is the phosphate group upside down. And so in this way, the 3' end is here, and the 5' end is here. It's upside down. Okay, they are going in opposite directions. So here is the bottom to the top, and over here, the bottom to the top is upside down. Okay, I'm going to draw another nucleotide, green. This one is bound to the sugar. So here's our sugar, and that's the phosphate group. And this is going to bind to a G. Okay, so I'm going to put the G here. It's upside down. And the G is a purine, so it has 2 rings. And the cytosine is a pyrimidine, it has 1 ring. Okay, so what I want to point out here is in between the DNA, so these rungs on the ladder has just enough space for 3 rings total. So a 2-ring purine always binds with a 1-ring pyrimidine. So you always have a purine with a pyrimidine. Together there is 3 rings. So in the middle of the DNA helix is enough room for 3 rings. So you always have a 2-ring with a 1 ring. Okay. So let's just add the last one. Let's see, I need another color, red. So this phosphate group is going to bind to this sugar, this deoxyribose sugar. And then it's going to, here's this phosphate group, and this is going to be a C, upside down C. And the C is a pyrimidine, which is 1 ring, and that's bound to a G, which is a 2 ring. And again, we just have 3 rings in the middle. And then so for the orientation, this is the 5' end, okay? So the DNA helix is always going in opposite orientations. If you know one side of the DNA, you automatically know the other side because A always goes with T, C always goes with G. So for example, if I were to write a sequence that said, let me just change colors to blue. If I said this is a DNA sequence that starts with a 5' and it's A, T, G, G, C, T, T, A, and that's the 3'. Oops. And I asked you what would be the complementary side of this DNA. You would automatically be able to tell me that the other side of this DNA is going to be 3', T, A, C, C, G, A, T, 5'. Okay? So if you know one side, you know the other side. Okay, so that's what you need to know for that. What else? Okay, 2 more things I want to go over. I want to go over the discovery of DNA. Okay, so a long, long time ago, this was in, I want to say the 1930, was it 1930 or 1950s? Hold on, let me think here. Yeah, 1953. Okay, was when DNA structure was discovered. Okay, so before we knew what DNA looked like and how it worked, people wondered and they knew that there was something being passed down from parent to offspring. Okay, so for example, you have these characteristics that are similar to your parents, like nose, like I have my dad's nose, and my skin color, I have my mom's skin color. And so you could see within a family that traits were being passed down from parent to offspring to the kids. And nobody knew how or what was being passed down exactly. Like, how is it that I have the same nose as my dad? Like, what exactly came from my dad and got into me? And so it took a long time, but people finally figured out that it was the DNA was being passed down from the parent to the offspring. Okay. And these traits were being passed down in the form of genes. Okay. So let me write that down. Genes are passed down from parent to offspring. From parent to offspring. Okay, and what this means is that you inherit a gene from your parents. So when something is passed down from parent to offspring, it's called inheritance. Okay, and what a gene is a gene is a section of DNA. So it's a section. It's a sequence of DNA nucleotides. So I'm going to write this down. Let me change colors just to make it in like a vocabulary word. So a gene is a section of DNA. It's normally about 1,000 nucleotides long. Section of DNA about 1,000 nucleotides long. And what it does is it is a code for a protein. So it's a code for a protein. Okay, so for example, I have brown eyes, and my brown eyes are brown because I have a code for a protein in my DNA, and it codes for a brown colored pigment. Okay, that is being expressed in my eyes. Okay, same is true for my skin color. I have a certain amount of pigment made because in my DNA there is a code for melanin protein. Okay, so your whole body and the way you look and the way you act and everything you are, and all living organisms, like a duck is a duck because it has all these codes in the DNA to make duck protein. We have all these codes that make human protein, and you look the way you look because you have your code. Okay, so that's what a gene is. And so people were figuring out what a gene was. We'll go into the history of that in another video, but I want to point out these three famous people who figured out the structure of DNA. Okay, so it was discovered by three people. And so during the 1950s, people like the scientific community figured out that DNA was being passed down from parent to offspring, but nobody knew what DNA looked like. And so it was kind of like a race. And all the scientists that were in this race knew that whoever figured out what DNA looked like could also figure out how it worked and could also win a Nobel Prize. And that's like the big, trophy for science is you want to win a Nobel Prize. So all these scientists just started trying frantically to figure out what does DNA look like and how does it work. And so all these different scientific people across the world were in this race to try to figure out what the structure of DNA was. And so there were two men. One, his name was James Watson. James Watson, and he's still alive today. And the other guy's name was Francis Crick. They were in England. They were very young. They were in their 20s, and they were working together on this project. Okay, and one of them came across a photograph from a woman. Her name was Rosalind Franklin. Rosalind Franklin was a scientist. And she did a lot of research on taking pictures of molecules. And molecules are very, very small. And so all she could do was take a picture of DNA using X-rays. Okay, so I'm going to show you an X-ray picture from Rosalind Franklin. She took a picture of DNA using X-ray, and all her pictures were using X-rays. And so this picture, Francis Crick and James Watson came across it, and based on this picture, they were then able to come up with how DNA, the structure of DNA. And the structure of DNA is what we drew earlier, that how nucleotides fit together. And based on this picture, they could see this is like looking down on the DNA helix from the top. And so you can see this X here, which gave them the hint that it was a double helix. And so, unfortunately, Rosalind Franklin died before the Nobel Prize was given out, and she died because she got cancer from working on X-rays all the time. So, X-rays are very dangerous, and they can give you cancer. And so, sadly, she didn't get the credit for the structure of DNA, but James Watson and Francis Crick did. But she is definitely in history, as being a contributor. Okay, so here is a picture of, up here on the top is Francis Crick, the bottom left is James Watson, and then on the bottom right is Rosalind Franklin. So all of them collaborating in a way, um, like gave us insight to what DNA looks like, and from then we could figure out how DNA looks and how it works. So I kind of want to show you a picture of a DNA molecule. Okay, so based on their discovery and hard work, they figured out what DNA looks like. And this is what we drew earlier. Okay, and so I just want to point out a couple of things. We have the 5' to 3'. Over here on the other side, it's in the opposite orientation. So 5' is on the bottom, 3' is on the top. And then each nucleotide joins with another nucleotide, and then each nucleotide joins across another nucleotide also. So here is the phosphate group. So let me circle it. So here's a phosphate group, here's the nitrogenous base. I'm sorry, that's the deoxyribose sugar, and then here's a nitrogenous base, and this is an A. And A is a purine, has 2 rings that binds across to a pyrimidine, which is T, because A always goes with T. And then here is the deoxyribose sugar, and here's the phosphate group. And so you join nucleotides going up and down, and then also across. And so in this way, they figured out that a DNA molecule is a ladder, and they figured out that it's twisted, and they figured that out based on this picture by Rosalind Franklin. Okay, so that's it for today's video. I hope you're having a great day, and I will see you next time.